This video is for Unit 3.2 on biological molecules. In this video I'm going to talk mostly about proteins. So as I've done in the other videos, we'll start by talking about the monomers or the building blocks of proteins. These are known as the amino acids. As we discussed in class, every amino acid has three parts. It has an amine group or amino group, and that's the one that has the nitrogen in it. It has a carboxyl group, or the carboxylic acid group. That's the part that has the double bonded oxygen in it. And then it's got this R group that joins the two of them together. And remember, this is the one that changes. This R could be many things. It could be a hydrogen, it could be CH3, and as you'll see on the next slide, there are a variety of different choices for that R group. When you built these in class, you had a similar structure here. Here's our amine group with the nitrogen with the two hydrogens on it. Here's the carboxylic acid group with a double bonded oxygen and then the O bonded to a hydrogen. And then in this particular R group, you had a carbon in the middle with three hydrogens. So you did see that structure in class as well. Remember, amino acids are just the building blocks, so we put many amino acids together to eventually make a protein. There are actually 20 amino acids. Some list 21 amino acids, some textbooks, but we will learn about 20 of them. And you'll be able to see in each of these diagrams here, all the different amino acids represented. They all have that amine group, that NH2. I don't need to circle all of them, but you can see that. And then they all have that carboxylic acid group. Okay. And then the thing that is different for all of them, the R group, is coming out the bottom. So you can see now the variety of different molecules that are bonded to that central carbon to create a different amino acid in each case because they have a different R group. All right, moving on with protein structure. If we link a bunch of amino acids together, each of these little circles representing a different amino acid, we bond them together into a structure called a polypeptide. So this whole chain, from beginning to end, is our polypeptide. Poly meaning many. So we have many amino acids put together to make that. This level of structure for a protein is primary structure of a protein, but we do not call this chain of amino acids a protein yet. It's a polypeptide. The next level for protein structure is called secondary structure. And as we did in class with the tubers, once you've got the polypeptide established, then you can bend it in different ways. The first way you can bend it is called an alpha helix. Sometimes alpha is written like that. The other way you can bend it is a beta pleated sheet. And sometimes beta is written like that. So this is just taking that polypeptide and creating hydrogen bonds to hold the structure. So in these little spaces between the polypeptide, it's being held together with hydrogen bonds to keep its secondary structure. Tertiary structure is when you take the beta pleated sheets and the alpha helices and you bend them even further. And this structure is held together by interactions between the R groups. This is, for example, in class, when you put the yellow push pins next to each other and they bonded each other, and you put the blue and purple next to each other and they bonded each other. Those are those interactions I'm talking about. There's all sorts of different kinds of bonds and rules kind of for how those R groups interact in tertiary structure. Another thing I wanted to point out to you is in one polypeptide you can have an area of alpha helices and then another area of a beta pleated sheet. So you can see in this example here, when the tertiary structure was being formed, here's an area that was an alpha helix, and here's an area that was a beta pleated sheet. And then that all got folded together because of those interactions between the R groups into a tertiary structure. Finally, the quaternary structure. This is when we put subunits of the tertiary structure together. So here you can see there's 1, 2, 3, 4 subunits. It's not always 4, but in this example it is. 4 subunits with that tertiary structure. So these are interacting with each other as well. There's bonds between the subunits. So we call, at this point, we can call this a protein. So the whole thing is a protein. But remember I said in class that some proteins also have that tertiary structure. So a protein could be a tertiary structure or a quaternary structure. This example just happens to be a quaternary structure. Alright, so let's look at it from the beginning again, just to make sure we have an overview of how this works. So we start with our primary structure down here. This represents each different amino acid. So this is tyrosine, lysine, alanine, valine, so forth and so on. Each of them are bonded together in a chain. Again, this is like the push pins that we made on the tubers in class. Then we can take that entire stretch of amino acids, that polypeptide, and we can fold it into a beta pleated sheet, or we can fold it into an alpha helix. That is called the secondary structure. Finally, we can take the tertiary structure, or the secondary structure, and fold it further. So here's some alpha helices that did have a beta pleated sheet, and now they're starting to fold in on each other. That's called our tertiary structure. And remember, tertiary structure could be a protein. And then finally, 2 tertiary subunits can start to bond together. 3 tertiary structures or subunits could, 4 tertiary subunits could. When 2 or more get together, basically we call that quaternary structure of a protein. Last thing we need to talk about is, why are proteins important? What do they do? The first thing that I want you to remember, and you went over this in class as well, is that the shape of a protein determines its function. So protein shape is really important. I just have 2 examples of some protein shapes down here. You can see that they're very different from each other. And if those shapes would change, they wouldn't be able to do what they're supposed to do. So what are some of the things that proteins do? A lot of proteins are used for structure, to build things. So to build your hair, to build your fingernails, to build the stretchy part of cartilage. So those are one of the main, or that is one of the main functions of a protein. Secondly, they are also used for repair. So because they're used to build things, when you have a something in your body that needs to be repaired, the proteins can build that back up. So they come and help out when you have damage in your body. Third, kind of related to that, is proteins kind of run the immune system. When we talked about the immune system in biology in 10th grade, talked about things like antibodies and antigens. They are proteins. Okay? Those things that help with cells identifying invading cells are proteins. And then finally, a big one that we're going to talk about in class next week is enzymes. Enzymes speed up chemical reactions. They are proteins, and we'll learn all about how they work next week. That's it for this video. Make sure that you fill out the form that lists the summary of this video as well as any questions you have for class.